Hello everyone, I am Amarika Bhalchandramue, pursuing Masters in Forensic Science and the volunteer at Applied Forensic Research Sciences. The topic we are going to discuss is the microcrystal test for drugs. Here is the content: introduction, procedure, examples, advantages, disadvantages, and references. Let's see the introduction. It acts as confirmatory test. In this microcrystalline test, on the basis of shape and color, the drug substance is identified. When we add specific reagent in drug sample, crystals are formed which are unique to that drug substance. The number and the size of crystals formed are variable according to temperature, pressure, hygrometry, or we can say humidity. This microcrystalline test is widely used to check whether the drug of interest is present or not in the sample. Simply, it reveals the identity of drug. Let's see the principle. It's a chemical test in which drug sample is combined with specific chemical reagent which results in formation of microcrystals that can be easily observed with the help of light microscope. It's a qualitative type test where only identification of drug is done. Let's see the procedure. A small quantity of drug is placed on glass side and few drops of chemical reagent is added. Stir it gently. A chemical reaction takes place resulting in formation of crystals. It is the shape and color of crystals which are unique characters for that specific drug. The results obtained with test reagent are then compared with references. Crystals are observed under light microscope. Now we will see some examples like cocaine, heroin, methadone, amphetamine, ketamine, phencyclidine are the common abuse drugs which can be identified using microcrystalline test. Let's see the example in detail. Suppose we need to analyze whether cocaine is present or not in the sample. For cocaine analysis, the reagent we need to make 5% aqueous solution. For that, add 1 gram of gold chloride in distilled water and make it up to 20 ml. Then for preparation of drug sample, add few microliters of acetic acid in drug sample. Further, in prepared drug sample, add few drops of reagent. Then egg-shaped crystal bisected by barbed rod, comb-like crosses and red one compensator is observed under microscope. This type of crystals are only formed in the presence of cocaine. Let's see advantages. It's cheap in cost, instrumentation is very simple, procedure is very fast, rapid results are observed, no need to extract sample. The reagent is required in very minute amount, the test is sensitive and rapid. The main disadvantages of this technique is that because of the reagent is used in it, the sample may be destroyed due to chemical reaction and the sample may be crucial, may be very in limited amount. The expert is required to perform and interpret the results in this test. These are the references. Thank you.